Hey everybody, in this video we are going to talk about gene regulation of the Lac Operon in prokaryotes. So please review the previous video where I go over the Lac Operon, and in this video specifically we are going to talk about regulation of the Lac Operon when glucose levels are low. So they're always low. And if you watched the last video, what that means is this cap cyclic AMP complex will always be bound to the promoter region. And RNA polymerase will always be able to be loaded onto the promoter, and the operator will be able to be transcribed. So just know that for this video, glucose levels are always low. This is always going to be bound to the promoter, so we don't need to worry about glucose levels anymore for this video. They're always low, and lactose levels are either going to be low or high. Okay, so before we go on, I need to introduce three different alleles: Lac OC, Lac I-, and Lac IS. Now, Lac OC, what this is, this refers to the operator region. And if a E. coli bacterium has the Lac OC mutation, what that means is that the region of the operator to which the Lac I repressor binds is mutated. So let's call this P, let's call this O, Z, Y, and A. Again, review the last video so you can see the general structure of the Lac Operon. Now, in the previous video, I didn't say whether or not these are wild type because they were wild type. We weren't talking about any mutant alleles at that point. But I'll put all pluses here to indicate that these are all the wild type sequences. But if OC is here, OC, C is for constitutive. Constitutive, meaning it's always on. The operon is always on. So, and what the mutation is you know how normally I draw the Lac I repressor binding site as a triangle? Now with the OC mutation, the sequence has been mutated. And so the Lac I repressor, it recognizes a certain sequence on the operator. And when that sequence is mutated, it can no longer bind to it. So the Lac I repressor can never bind this. It doesn't matter if lactose is high or lactose is low, it's never going to bind here. As a result, RNA polymerase is always going to be able to load on the promoter. Why? Because, and remember in the beginning of the video, I said we're talking about low glucose conditions. So cyclic AMP cap complex, that's always bound to the promoter. And it's always helping RNA polymerase get onto the promoter. And because the operator binding site, the binding site on the operator for the Lac repressor is mutated, well, this is always going to be transcribed. RNA polymerase is never going to be blocked. It's always going to be able to make the polycistronic RNA. So another one, Lac I-. Now there are two possibilities for Lac I-. We can have a Lac I- allele that is just a null allele. What does that mean? It can't make a functional protein. It does not encode a functional protein. So there's just no Lac I repressor anymore. If there's no repressor, then the repressor can never bind to the operator. It can never block RNA polymerase. The operon should always be transcribed. Now, I should be careful. I'm saying always and never a lot, right? So we're going to look at these in different situations. We're going to look at these when they're on plasmids or when they're on the chromosome, and then we might see some strange things going on. So I said there could be two possibilities here. One is it's a null allele. Another is just let's see, how would I diagram this? Is there's been a mutation in the binding site, in the operator binding site. So if this is the Lac repressor, and we know how when lactose levels are high, the Lac repressor looks something like this, right? Because lactose is bound to it. That operator binding site disappears. The binding site for the operator disappears. Now, a Lac I- allele would mean that even when there's no lactose, that site is still gone, still not there. So this is essentially equivalent to a null allele, right? Because you get the same thing. You get a non-functional protein. This protein is supposed to bind the operator when lactose levels are low. But we're seeing, because of this mutation, it's no longer able to do that. So this is a non-functional protein. But to keep things simple, I'm going to try and just, throughout the rest of this video, assume the Lac I- is a null allele. So the protein, it's not there. It's just not there anymore. Maybe this mutation means the start codon or something is mutated to a stop codon. So the bacterium doesn't make this protein. Protein's not there, can't bind the operator. The operon is transcribed. Okay, so this, Lac IS, this is known as the super repressor. So what does it mean? So maybe I'll try and edit this video here, right here, so as not to not waste your time. Okay, start over because it's been here a long time. So Lac IS. So Lac IS, this is known as the super repressor. And what does this mean? So what is a super repressor? So essentially, let me diagram this for you here. So the super repressor looks something like this. So here is low lactose conditions, and here is high lactose. And why is it high lactose? Because look, there's a lactose here. But with Lac IS, there has been a mutation such that the lactose binding site is gone. So lactose can no longer bind to the Lac IS, to the Lac I protein. If lactose can't bind, it can't go through that allosteric shift to eliminate the operator binding site, right? So this is always going to be bound to the operator, and it's always going to block RNA polymerase, and the operon is not going to be transcribed. So that's what the Lac IS mutation does. It shuts down expression of the Lac operon. So that E. coli would be in pretty big trouble, right? If its only carbon source was lactose. So we have a couple different situations there, a couple different alleles. So what I'm going to try and do now is go over some problems that you might encounter on an exam, or, well, I guess we don't have quizzes left for this section of the course. We do have an exam though. And I have a diagram here, and I hope it looks like you can see it pretty well. You may need to zoom in a little bit, or definitely watch this on a computer, not your phone. So with this diagram right here, what am I showing you? First, down here, what is this thing? Like I said before, this is the cyclic AMP cap complex. And for this whole video, we're going to assume glucose is low. And when glucose is low, the cyclic AMP cap complex is bound to the promoter. So this is the promoter region. This is the operator let's say these are all plus for now. Z plus, Y plus, A plus. So to see what all this glucose is low and C cyclic AMP complex, what this is about, make sure you check out the last video. But glucose is low, this is bound here, RNA polymerase can bind to the promoter and transcribe the operon. So what is all this stuff here? So what I'm showing you here is that this is the E. coli chromosome, and let's imagine we're in an E. coli cell right here. And here, what I'm showing you is, okay, we're zooming in. So the chromosome is 4.6 million base pairs long. And here we're zooming in. I'm just showing you one gene right here. And this is going to be Lac I. Now, if you remember, Lac I is not part of the operon. It's expressed from its own promoter from somewhere else in the genome. So this is the Lac I gene. Now, over here, now this part of the chromosome, now this is where we're going to say the Lac Operon is. And here we have the promoter, the operator. I should put this little triangle in there, right? This is O+. This is the Lac repressor binding site. Lac Z, Lac Y, Lac A. And what is this over here? So for these problems, we're going to take a look not only at the chromosome, but a plasmid that might exist in E. coli. This is the F' plasmid. In the first section of the course, we looked at F+, right, and F- strains, and we're going to call this plasmid F', and we don't need to worry too much about why we're calling it F', but we just need to know that it's a plasmid. And we know E. coli can have plasmids, and we can put genes on these plasmids to do different genetic tests and to see how these genes work when multiple genes involved in lactose metabolism are present in the bacterium, on the chromosome, and on the plasmid. And again, this is zooming in. The plasmid is much smaller. I didn't look this up. I'm just assuming it's about 15 kb. I did look this up. 4.6 million base pairs is accurate. This, I'm guessing about 15 kb would probably be long enough to do these types of tests that we're going to talk about. And we're going to put a gene here. So I'm zooming in and showing you, okay, there'd be a gene here, and we're going to be interested in the identity of this gene. Now, so let's assume that the genotype of this E. coli is Z+, I+, O+, and we won't worry about any like Y and A right here, or the promoter. We won't worry about that. Just Z+, I+, and O+. Okay, everything's wild type here. Now, so what would you predict would happen under high lactose conditions? And then we'll take a look at low lactose in a moment. So under high lactose conditions, okay, so another thing to keep in mind, Lac I, this is always expressed. So if Lac I is wild type, I'm going to say Lac I+, right? Genotype is Lac I+. Let me put genotype up here. Genotype is Lac I+. So we're always going to have, the cell's always going to have the Lac I protein available to bind to here. Now when lactose is high, right, what's going to happen? Lactose will be bound to Lac I. I'm outside of the cell here. Sorry about that. This would be in the cytoplasm. And I diagram this like really in there. That lactose is really inside the Lac I repressor. But you get the point. When that's bound here, oh, okay. We're going to lose that binding site. The allosteric shift occurs. This can no longer bind the operator. Lactose is bound. So RNA polymerase will transcribe the operon. So operon is transcribed. What about when lactose is low? Lactose is low. This is not bound here. So we'll just have this. This will bind here, and it will block transcription. So operon not transcribed. Now in this problem, we weren't even concerned with the plasmid there. So let's say there was a plasmid there, but maybe there was no genes that were involved in Lac Operon regulation. Okay, so I think we understand that. So now we're ready for a trickier problem. So let's assume the genotype is Z plus I plus OC. Okay, so again, this is P plus OC. Well, this is OC, so there's no binding site anymore. You know what? I'm going to mess it up. So there's a mutation there. That binding site is no longer recognized by the repressor. Z plus Y plus A plus. Still not worried about the plasmid just yet. This is I plus. Okay, so what about high lactose? And low lactose. Okay, we'll take high lactose first. I should have diagrammed the Lac repressor on here on the template. Okay, we are high lactose. This is bound. Lactose is bound. As a result, the operator binding site is gone. So under high lactose conditions, it's going to be transcribed. RNA polymerase is going to bind, nothing's blocking it, transcribed. Operon transcribed. What about low lactose? Okay, so low lactose. Low lactose. Lactose cannot bind here. This is open. It can bind the operator, but the operator site has been mutated, right? This is the OC mutation. So it can't bind, it can't block RNA polymerase. So even when lactose is low, the operon is transcribed. Operon transcribed. You can say, yes, operon transcribed. Yes, operon transcribed. Okay, so now a twist. What if the genotype is the same here for the chromosome, but now there's something on the F plasmid? A wild type version of the operator. So this section right here, we just take it and we put it on this plasmid over here. So I'm going to say this is O plus. So it has the functional, the wild type binding site for the Lac repressor. So when lactose is low, Lac repressor is here, it's going to bind over here. So will that block transcription of the operon when lactose is low? Lac I, it's functional. No lactose is bound, so the operator binding site is open. Can't bind to the OC region on the operon in the chromosome, but it can bind over here. So will the operon be transcribed? And what you do, you need to think about how this functions, right? It physically blocks transcription on the operon. It needs to be present in order to block RNA polymerase, whether it's a physical blocking or it just obstructs the promoter binding site. You know, it doesn't really matter. But it has to be on the operon in order to block transcription. It's way over here on the plasmid, it's not going to block anything. So just because there's an O plus on the plasmid doesn't mean the operon with the OC region can now be properly regulated. This will have no effect. So when lactose is low, the operon will be transcribed even when O plus is on the plasmid. If the operon has the OC mutation. So what we would need to do, we could look at the other way, right? So what if O plus was over here? What if O plus was here and OC was over here, right? I wonder if I can erase. It'll probably be easier than... Where did my eraser go? Here it is. Okay, yeah. So we put O plus over here, and we put the mutated operator over here. So I'm going to erase this for now. It gets confusing going back and forth, I bet, doesn't it? I mean, it gets confusing for me, so I imagine it must for you also. So, okay. Now we moved O plus over here, we put the OC over here. This guy, low lactose, no lactose bound here, it can bind, it can block. So when lactose is low, RNA polymerase cannot proceed to transcribe the operon. It doesn't matter that LAC-I cannot bind here, because this has, OC, it has no effect on regulation of the operon because it needs to be over here on the operon. Just because you put a copy of this non-functional operator on a plasmid located far away from the operon doesn't mean RNA polymerase is going to be able to transcribe the operon uninhibitedly, if that's a word. So does that make sense? I think that makes sense. So and the reason we talk about this is, when we say OC, okay, that means the operon's always going to be transcribed. And no, not necessarily. It means where it's located influences whether or not it has effect on transcription of the Lac Operon. It has to be located within the operon in order to have an effect on the operon. Now, I just want to take a look at one more, which can be confusing. Now, the notes has many, there are many problems in the notes related to this type of setup right here, with different genes here, different mutations here. So I encourage you to take a look at those. But let's just do one more in this video. And the videos are kind of hard to make. I don't really have time to do multiple takes here. So I hope this turns out to be helpful for all of you. So let's look at a genotype that is, say, Z plus, I plus, O plus, with the super repressor on the plasmid. So over here, we have the super repressor, LAC-IS. Over here, we have LAC-I plus. And we have the wild type promoter, wild type operator, Z plus, Y plus, and A plus, which is what you all want to get in this class, I bet, right? No, but we don't do A pluses here at Illinois State University. So operator is wild type, so the operator binding site is normal. Now this LAC-I is normal, so we're always going to have some normal or functional LAC-I protein in the cell, but we're also going to have the non-functional, well, not functional, non-functional, the super repressor which has the mutated lactose binding site. And cyclic AMP cap is bound here, so RNA polymerase is going to have no trouble binding to the promoter. But will it be able to transcribe the operon? It depends whether or not there's something here. So let's consider let's consider high lactose first. High lactose conditions. So under high lactose conditions, will this operon be transcribed? Well, if lactose is high, we can put this here. I'm sorry, this was I diagrammed this incorrectly, didn't I? When lactose is bound, no, I diagrammed it correctly. When lactose is bound, the binding site disappears. The binding site for the operator disappears. I don't know why I'm coloring this in. And it can no longer bind here. Okay, so RNA polymerase can transcribe, right? There's nothing blocking it. But wait. There's lactose here, but it can't bind to the Lac-I superrepressor. The Lac-I superrepressor is always going to be bound. So it's always going to be blocking transcription of the operon. So in this case, look, it doesn't matter that there's a functional copy of Lac-I plus here. The Lac-I superrepressor, you could say in this case that Lac-I superrepressor is dominant. You know, it's dominant over Lac-I plus because it's here. It doesn't matter whether lactose is high or low, it's always going to be bound here. This could be functional. Lac-I plus is functional, but it has no effect because Lac-I superrepressor is always bound. So under high lactose conditions, RNA polymerase will be blocked. It cannot be transcribed. And even under low lactose conditions, also, it's going to be bound there too, and it won't be transcribed. So there were a couple of examples there. What are some others? I think you can move like Lac-I minus over here and I plus over here. What effect does that have? You can look at Z minus, and you know it'll still be transcribed, but beta-galactosidase won't be functional. There are a couple other examples. I think I'm blanking on them right now, but they're in the notes. Make sure, and I typed it up nice and neat in the notes for you so you can look through it. I think you should understand how these types of questions work and be able to answer them on an exam now. But if not, if you're still confused, make sure to send me an email, hopefully before the night before the exam. And I'll try and help you out, or maybe we'll remake this video and try to clarify some things. Okay, see you in lecture 18. Okay.